So, good morning to all of you students of BT First Sim. Today, I'm going to discuss with you about the nucleus structure and its function that is given in your cell biology syllabus. So, let's get started now. So, generally, this is the ultrastructure of a nucleus, and according to if I say through introduction, the cell nucleus is the most noticeable organelle within the eukaryotic cell, and perhaps it is the most important and defining feature of eukaryotic cells. Most of the genetic material, that is your DNA, is contained within your nucleus, while a small amount of DNA is found within the mitochondria, as we have seen, which is called as mtDNA or mitochondrial DNA. Most of the human cell have a single nucleus, but there are some cell types which have multiple nuclei. Plural form of nucleus is nuclei, like in osteoclasts. So in osteoclasts, they have multiple nucleus. I'm writing in good way now. Let us write osteoclast. OSTEOCLASTS. So they have multiple nuclei, and some of the cells don't have nucleus at all, like in erythrocytes, the red blood cells. They don't have any nuclei. Generally, nucleus is about your 5 to 10 nanometer, micrometer in diameter, and it is found in multicellular organisms. Most of them multicellular organisms have nuclei, and it is the largest organelle that is found within the cell. That's why it is a noticeable structure. The smallest nuclei is approximately 1 micrometer in diameter and is found in the yeast cell. Now, most of the shape of the nucleus is either spherical in shape or oblong in shape. Well, oblong means it somewhat look like oval shape like this, or spherical in shape. So here you can see in the diagram a oblong shaped nuclei has been shown, right? And usually cells have one nucleus, but at times there may be multinucleated cell as well. And why multinucleus occur within some cells like in osteoclasts? It's due to karyokinesis. That means when your cell undergoes nuclear division, during that phase, karyokinesis, during your karyokinesis, which is the division of the nucleus, okay, during cell division, your multiple nuclei may occur, or sometimes the cell fuse. Sometimes the cell fuse to form syncytium. Syncytium, not cytium, syncytium. Multiple cells can fuse to form syncytium, like in mature muscle cells, which can be observed. And due to this fusion, multiple nucleus can also be observed. So, two reasons are there why multiple nucleus can be observed. It's due to the cell division process during karyokinesis, when the nucleus undergoes division, then multiple nuclei may be observed, and during syncytium, where multiple cells fuse, at that time also you can see multiple nucleus. Well, we can also see multiple nucleus when we fuse two protoplasts, two cells, to protoplast, divide of cell wall, removing of cell wall, they fuse together to form a heterokaryon having multiple nuclei. That can also be done. Okay. Now, nucleus has very important role to play, as we are seeing here. It is one of the vital cell organelle. It contains the genetic material, right? It also coordinates the cell activities like your protein synthesis, like your cell division. Okay, so it has many vital functions to do. And anatomically, if I see, nucleus is made up of several components. As you can see here, nucleus is made up of many components like your nuclear envelope, okay. The nuclear envelope is composed of outer nuclear membrane and inner nuclear membrane. Together forms the nuclear envelope. As you can see in this diagram, I have marked it. It is also, nucleus is also made up of nuclear lamina. You can see here the inner nuclear lamina has been mentioned here, right there. You can see, if I zoom it in, you can see the inner lamina here, stripe type of figure. This is the inner lamina of nucleus. It is also composed of your nucleolus, where you can see the pinkish here. You can see here the nucleolus, right, and the chromatin fibers, okay, which will form chromosomes when they will uh, closely associated with the histone and the non-histone proteins. The chromatin fibers, these are the chromatin fibers which are widespread throughout them, okay. And your nucleoplasm, which is the cytoplasmic part of the nucleus. Well, between the nuclear membrane, you can also see some gaps here. You can see here, if I zoom it in, you can also see a nuclear pore is observed between the nuclear membranes. Okay, so I marked it. I have in two sides. I have marked. In this way, there are many multiple nuclear pores through which your specific function, like removal out of the uh, to remove out the mRNA for the purpose of protein synthesis. Okay, then other things are also being observed. Okay, means transportation is done through this nuclear pore complex, you can say. Okay, you can also see some intermediate filaments are forming a cradle like structure within the nucleus. Okay, as you can see here, some intermediate filaments. So all of these components, whether you are seeing here nuclear envelope or the nuclear lamella, the nuclear pore, the nucleoplasm, the chromatin thread, the nucleolus, all of these components work together in order for the nucleus to accomplish all of its functions. And some of these functions are, if I say in point wise, that is given in your notes. First of them, it control the genetic information of the cell, and thus the heredity characteristics of an organism is regulated by this nucleus. Okay, it also control the protein and enzyme synthesis. Third, it controls the cell division and cell growth, because without karyokinesis, your nuclear division will not occur. Cell division is consists of two phases. One, we know the prophase, metaphase, anaphase, telophase, but if I say broadly, two things mainly, or two events mainly occur. One is karyokinesis, one is cytokinesis. So it also controls the cell division and cell growth. It stores the genetic information in the form of DNA, in the form of RNA and ribosome. Yes, RNA synthesis occur within this nucleus. The transcription process occur within this nucleus. Also, the ribosome synthesis occur within the nucleus also. It regulates the transcription of the mRNA into protein and production of ribosomes. Okay. Now, in the laboratory, if I stain the nucleus with some particular dye with particular stain, it can able to absorb the strain, and we can able to see the nucleus as a dark organelle. Okay, so let us now discuss one by one about the structure of the nucleus in detail. So let us discuss about nuclear envelope. Nuclear envelope, also called as perinuclear cisterna, cisterna, that encloses the DNA and defines the nuclear compartment of interphase and prophase nuclei. Again, I repeat, your nuclear envelope, or it is also called as perinuclear cisterna. This nuclear envelope actually it encloses your DNA and defines the nuclear compartment of interphase and prophase nuclei. It is formed of two concentric unit membrane. One is your inner nuclear membrane and is outer nuclear membrane. The inner nuclear membrane contains specific proteins. If you see the inner nuclear membrane, it contains specific proteins that will act as binding site for the supporting intermediate filaments. Okay, and that is called as nuclear lamina. So again I repeat, the spherical inner nuclear membrane. This spherical inner nuclear membrane contains specific proteins that will act as binding sites for supporting the fibrous set of your intermediate filaments, and that is called as nuclear lamina. Okay, the inner nuclear lamina, if I say the inner nuclear lamina, they are actually internally lined protein filament meshwork which is organized in a net like fashion, right? And they are interacting with the inner nuclear membrane. If I show the diagram more more easily diagrammatic view, it has been given in your notes. I am showing you. Wait. So here you can see the nuclear lamina, which are actually internally lined by protein filaments meshwork, and it organized like a net like fashion. Okay, and the proteins actually which make this nuclear lamina are called as lamines. Okay, are called as lamines, and actually lamines are a type of intermediate filament proteins. This nuclear lamina is actually supporting your nuclear envelope and ensuring that the overall shape and structure of the nucleus will be maintained. So again I repeat, nuclear lamina is actually what? It is actually made up of lamine intermediate filaments which are supporting the nuclear envelope structure to ensure the overall shape and structure of the nucleus can be maintained. Okay, there are also some lamina associated proteins which cannot be represented in this diagram. I have shown a simple diagram of the nuclear lamina. Okay, other proteins are also there. Some additional proteins are also there called as lamina associated proteins, which helps to mediate the interaction between the nuclear lamina with the inner nuclear membrane. Okay, so the inner nuclear membrane is interacting with this nuclear lamina. That's why I have a little bit describe about this nuclear lamina. Okay, and it is made up of laminin proteins with lamina associated proteins, which helps to mediate the interaction between the lamina and inner nuclear membrane, and also they are supporting the nuclear envelope and to maintain the overall shape and structure of the nucleus. Okay, so we were doing about the nuclear membrane, the nuclear envelope. Now, the inner nuclear membrane, if I see, it is surrounded by the outer nuclear membrane, which closely resembles like the membranes that is found within the endoplasmic reticulum, that is continuous with it. That means the outer nuclear membrane is continuous with the membrane found in the endoplasmic reticulum. Like the membranes of the rough endoplasmic reticulum, outer surface of the outer nuclear membrane is decorated with the ribosomes, which are engaged in the protein synthesis. Again, I repeat, the surface of the outer nuclear membrane is decorated or bounded by ribosomes on its surface, which are engaged in protein synthesis, and it is continuous, right? It is continuous. Okay, from the nuclear outer membrane, it is continuous with the outer membrane of the of the endoplasmic reticulum. Now the proteins made on these ribosomes, the ribosomes are making proteins on the outer nuclear membrane. Okay, so the proteins that made on these ribosomes are transported into the space between the inner and the outer nuclear membrane, and that is called as interperinuclear space. Okay, again I repeat, I have said about the inner nuclear membrane first. The inner nuclear membrane contains specific proteins that interact with the intermediate filaments called lamins, which form the nuclear lamina, right? To maintain the shape and structure of the nucleus and also to interact. There are also some other intermediate filament proteins called lamina associated proteins also, which helps to mediate the interaction between the nuclear lamina and inner nuclear membrane. What about the outer nuclear membrane? The inner nuclear membrane is surrounded by the outer nuclear membrane. Okay, it is surrounded by the outer nuclear membrane. It just looks like this. If I show you in another diagram, it will look like this. Here you can see the inner nuclear membrane is continuous with the outer nuclear membrane, right? And it is like the membranes of the rough endoplasmic reticulum. The outer nuclear membrane is also decorated with ribosomes, which are engaged in protein synthesis. Now, the proteins which are made on this outer nuclear membrane is transported between this space. Okay, it is transported between this space. So you can see the space here. So the proteins which are synthesized by the ribosomes on the outer nuclear membrane, suppose these are the ribosomes. So, and if I say these are protein being synthesized, so they are getting within the space between the inner and outer nuclear membrane after it is being synthesized. That is called as perinuclear space. Now this perinuclear space is about 10 to 50 nanometer in wide, 10 to 50 nanometer in wide, which is filled with fluid which which is continuous with the endoplasmic reticulum lumen, right? That means the proteins enter to this perinuclear space, but it is continuous with the rough endoplasmic reticulum. Suppose if this is not present, then it is continuous with the uh, membranes of the rough endoplasmic reticulum. Okay, suppose it's the rough endoplasmic reticulum, so it enters in this way, and it is secreted, just like I have said how the secretory proteins are getting modified, packaged into vesicles, from the rough endoplasmic reticulum to the Golgi apparatus. Okay, so this was about the your nuclear envelope, which is made up of inner and outer nuclear membrane. Now about the nuclear lamina, I have already discussed with you that nuclear lamina is actually what? They are actually a protein meshwork which is of about 50 to 80 nanometers thick. So this nuclear lamina, as it is shown in different diagrams showing different things. Here the red lines has been showing by the nuclear lamina to represent nuclear lamina. So nuclear lamina are what? They are made up of intermediate filaments called lamins. And generally three types of lamins are going to be observed here: lamin A, lamin B, lamin C. And these lamins that are making your nuclear lamina, they form dimer like structure, and they have a rod like domain with two globular heads at one end. Okay, so these lamins are mainly making your nuclear lamina, and as I'm repeatedly saying, they are supporting the nuclear envelope and they are maintaining the overall shape and structure of the nucleus. Additionally, these nuclear lamina also contain some lamina associated proteins which helps to mediate that interaction with the inner nuclear membrane. Okay, okay, so let us move to the next part, next thing, which I will going to discuss now. Okay, so now we will discuss about the nuclear pore, which is showing right here. Okay, it has been mentioned here. If you see this nuclear pore complex, it is showing just like a tube-like structure, but it is little bit more critical in shape. If I show you the diagram, okay. So let's move to the next part of the nucleus, the component of the nucleus, which is your nuclear pore complex. So let us discuss about the nuclear pore complex that is found within the gap between the nuclear membranes, between the nuclear envelopes. So as you can see in the diagram, on the left diagram, now it looks like a toy, right? But it's a in this way nuclear pore complex is observed actually. Nuclear pore appears circular in view and have a diameter between 110 nanometer to 100 nanometer. So from 10 to 100 nanometer is the diameter of this whole nuclear pore complex. Now this pore complex, as you can see in this diagram, the three-dimensional diagram, it is composed of two rings. If I zoom it in, you can able to see. So it is composed of two rings, one ring right here and another ring right here. Okay, so it is composed of two rings. I'm showing in different colors so that you can able to see it. So it is composed of two rings, one rings right here and another rings right here. As it is showing in three-dimensional view, you can able to see it more clearly. Now these two rings are also called as annuli. They are called as annuli. Okay, or annulus. Okay, so these two rings are away from the center. They are away from the center of this nuclear pore complex and has a diameter of about 80 nanometer. There is a central plug that you can also see. There is a center plug that you can also see, and radial spokes. These are the radial spokes, okay, that extend from plugs to the rings. So it joins the plugs to the rings. So you can see radial spokes right here. Okay, the spokes are this one, these are the spokes. Okay, More easy diagrammatic view has been shown. You will see them. So there are radial spokes right here, which join to the uh, plug to the rings. Okay, this plug will be acting as a transporting agent. They will be helping to transport the molecules out and in from the nucleus. Okay, now the hole in the center of the nuclear pore complex is a channel. It is an aqueous channel through which water soluble molecules will shuttle between the nucleus and the cytoplasm. Okay, there is a central plug through which it able to bind and prevents the movement of the molecules. So it selectively allows movement of the molecules across the nucleus, right? So now I am going to show you the main more proper diagram, more appropriate diagram of the nuclear pore complex. So this is the more appropriate diagram of the nuclear pore complex. This is your upper view. From upside view you can see the central plug, okay, and the two rings has been shown, right, this one and the outer ring like this one. Okay, these are actually what? These are actually the ribosomes which are being attached to the nuclear filaments. Okay, these are the ribosomes which are attached to the nuclear pore complex, to the rings or annulus. The spoke which helps to join the plug with these rings. Okay, so as I have said that this nuclear pore complex composed of two rings, also called as annuli, at its periphery. Periphery means away from the center. The rings are away from the center of the nuclear pores and have a diameter of 80 nanometer. And it has a central plug also. This central plug are joined with joined with the rings or annulus or annuli with the help of spokes that extend from plugs to the rings. Then the hole in the center that you can see where the central plug is there, the hole in the center of the nucleus pore complex is an aqueous channel through which water soluble molecule will pass between the nucleus and the cytoplasm, and this hole often appears to be plugged with this protein by a large central granule called central plug, which believed to consist newly made ribosomes and other particles caught in the transit. Okay, so it's just appear like this. Sideway it appears like this. You can see more easy diagram has been shown. So the central plug is actually it is a granule. It is not a protein. It is a granule which is believed to consist of newly made ribosomes or other particles caught in the transit. Now this nuclear pore complex are they perforates the nuclear envelope, bringing the lipid layers of the inner and the outer nuclear membrane together around the margins of each pore. So you can see this is the nuclear envelope of its inner and nuclear outer membrane. They merge at a point, and this is the space where this nuclear pore complex is observed. Okay, now within the nucleus of mammals, it has been calculated that this type of nuclear pore account for 5 to 15% of the surface area of the nuclear membrane. Like in amphibians, it's different. Nuclear pore complex composed of 20 to 36% of the surface area within the nucleus. So in amphibians, it contains much high amount of nuclear pores compared to the mammals, which contain only 5 to 15% of the area surrounded by these nuclear pores. And they have actually this number of pores in the nuclear envelope correlate with the transcriptional activity of the cell, actually, you can say. Now, more if I say more specifically about its function, about this nuclear pore, is that within this nuclear pore there will be movement of the molecules across the membrane from nucleus to the cytoplasm and vice versa. Okay. Now, quite evidently, if I say, there is a considerable trafficking across this nuclear envelope during your interface, like your ions, nucleotides, structural, catalytic, and regulatory proteins are imported from cytosol to the nucleus. Okay, the mRNA, tRNA, and ribosome subunits are exported out from the nucleus to the cytosol. So, what type of molecules are entering and removing out from the nucleus with the help of this nuclear pore complex? Ions, nucleotides, structural, catalytic, and regulatory proteins are imported from cytosol to the nucleus, and your mRNA, tRNA, and ribosomal subunit are exporting out from nucleus to the cytosol. However, one of the main function of this nuclear um, pore complex is to however, the main function of the nuclear envelope is actually to prevent the entrance of active ribosomes to the nucleus. So here the active ribosomes are prevented to enter to the nucleus, and that is prevented by this nuclear pore complex. Okay, so this nuclear pore complex is preventing the entry of active ribosome because its function is outside now. Okay, and it is selectively allowing the entry of mRNA, tRNA, and ribosomal subunit to exported out to the from the nucleus to the cytoplasmic matrix, that is to the cytosol, and it is allowing the entry of the ions, nucleotides, and structural catalytic regulatory proteins to be imported from cytosol to the nucleus. So the nuclear pore complex appear to be function like a close fitting diaphragm that opens to just right extent when it is activated by a signal on an appropriate large protein. So I have said the central plug, right? I have said the central plug. There must be some signal mechanism going on, okay, which helps to open that plug to be allowing entry of the particular specific molecules towards the nucleus and removing out from the nucleus. Recently, it has been investigated that there are some specific nuclear proteins. There are some specific nuclear proteins called karyophilic proteins. Karyophilic proteins which have molecular structure like signals. These proteins will maintain a signals, okay, or nuclear import signals that enable them to accumulate selectively in the nucleus. Again, I repeat, recently studied had been done that investigated that there are some proteins called cardiophilic proteins which mediate cardiophilic signals. So these cardiophilic proteins will mediate some signals. Okay. Or you can say nuclear import signals which enable them to accumulate selectively in the nucleus. This cardiophilic signal for nuclear import apparently resides in the tail domain, and such an active nuclear transport require energy which is derived from ATP hydrolysis. So there will be an active transport going on. This cardiophilic signal will trigger that event. Okay. So in this way, your nuclear pore complex works within the nucleus, Okay. Now let us discuss about your nucleoplasm. Well, nucleoplasm, as you can see, it is the just like a cytoplasm that is found within the cell. Nucleoplasm is somewhat similar to cytoplasm of a cell, which is a semi-liquid, and it fills the empty space within the nucleus. Now, this nucleoplasm is formed. It is a form of protoplasm which surrounds the chromosome and nuclei inside the nucleus, and various type of proteins and enzymes dissolve within this fluid, dissolve within this nucleoplasm. Some nuclear bodies are able to observe within this nucleoplasm, like your specific structure like Kagel bodies, CAJAL, Kagel bodies, Gemini bodies, Gemini bodies, and polycomb bodies are observed. So within this nucleoplasm, we can able to observe such type of nuclear bodies. Kagel bodies, if I say, Kagel bodies are the regions within the nucleus that are enriched in proteins and RNAs involved in mRNA processing. Again, I repeat, Kagel bodies, which I have mentioned here, these Kagel bodies that is found within the nucleoplasm. These are the regions, Kagel bodies are the regions that is found within the nucleoplasm. They are enriched with proteins. Kagel bodies are enriched with proteins and RNA, which will be involved in which will be involved in the RNA processing, specifically the mRNA processing. And for mRNA processing, we need spliceosome complex to remove away the introns from the exons to make a mature mRNA. And this whole spliceosome complex is made up of small nuclear ribonucleoproteins that is found within these Kagel bodies. So Kagel bodies are actually the main site for assembly of the small nuclear ribonucleoproteins, also called as snRNP, small nuclear ribonucleoproteins. These small nuclear ribonucleoproteins will make your spliceosome complex, will make your spliceosome complex, which will catalyze the splicing process, where the introns of the mRNA will be removed out, the non-coding regions will be removed out, and the coding regions, that is the exons, will be joined to form a functional mature mRNA. So this is the main function of Kagel bodies. Kagel bodies are the main sites for assembly of the your small nuclear ribonucleoproteins. Okay, they are the sites for assembly of the snRNP. Okay, and Kagel bodies are also called as coiled bodies. They have a spherical nuclear bodies of about 0.3 to 1 micrometer in diameter, and it is found within the nucleoplasm of embryonic cell, those cells which are proliferative actually. Okay, like in embryonic cells, in tumor cells, or metabolically active cells like neurons. Okay. So these causal bodies which are found within the nucleoplasm, uh, as I am saying, they are the sites for assembly of snRNP. They are normally found in proliferative cells, which are constantly proliferating, active cells like embryonic cells, like tumor cells. So within the nucleolus of embryonic cells, within the nucleoplasm of embryonic cells, tumor cells, and metabolically active cells like neurons, they have causal bodies. Causal bodies are membraneless organelles. They don't have any membranes and largely consist of proteins and RNA. Causal bodies have been implicated in RNA related metabolic process like maturation of the RNA, recycling of the small nuclear ribonucleoprotein, then histone mRNA processing, okay, and the telomere of the chromosome that we know, the maintenance of the telomeric region can be done by the causal bodies. Causal bodies are assemble RNA, which is used by the telomerase enzyme to add nucleotides at the end of the telomere. Again, I repeat, causal bodies assemble the RNA which is used by the telomerase enzyme to add nucleotides at the end of the telomeres. Telomerase enzyme catalyze the formation of telomere regions which will protect the ends of the chromosomes. So, vast function found by the causal bodies, which are the small bodies found within the nucleoplasm of the proliferative cells, like your tumor cells, your embryonic cells, and metabolically active cells like neurons. Then, within the nucleoplasm, we also find Gemini bodies. Also, Gemini bodies are also nuclear bodies, which are frequently and found to be close associate with the causal bodies actually. Okay, Gemini bodies are of similar in size and shape with the causal bodies, but it is different from the causal bodies because uh, Gemini bodies do not contain the small nuclear ribonucleoproteins. Okay, Gemini bodies, they closely associate with the causal bodies within the nucleoplasm, but the main distinguishable feature is that these Gemini bodies don't contain the small nuclear ribonucleoproteins. That means they don't have this snRNP. Instead, they have a protein called survivor of motor neurons. Survivor of motor neurons, also called as SMN, survival of motor neurons, whose function is related with biogenesis of snRNP. So they are involved in biogenesis of small nuclear ribonucleoprotein, whereas the causal bodies are involved in what? Causal bodies are involved in assembly. They involve in assembly of these small nuclear ribonucleoproteins. So for biogenesis of the snRNP, for biogenesis of snRNP, for the biogenesis of small nuclear ribonucleoprotein, your Gemini bodies are involved, whereas for assembly of snRNP, your causal bodies or CB are involved. Right? Okay. So Gemini bodies that is found within this nucleoplasm, the diagram has not been shown the Gemini bodies. Just for your BSc syllabus, you have to learn this much only. The Gemini bodies that is found within the nucleolus, they contain SMN proteins called survivor of motor neurons, which participate in biogenesis of your snRNP, which is involved in the biogenesis of small nuclear ribonucleoprotein biogenesis. Gemini bodies, they believe to assist the Kaggle bodies in snRNP biogenesis, and they also play important role in the etiology of spinal muscular atrophy. Okay. Another bodies which is found within this nucleoplasm are polycomb bodies, polycomb proteins, better to say, polycomb. Polycomb bodies also play an important role in gene silencing. Gene silencing is a process where we regulate the expression of the gene through different mechanisms. Either at the transcription level, before translation, we can also silence the gene. Okay, so polycomb proteins also found within the nucleoplasm, and they play an important role in gene silencing through different mechanisms. These protein act in complexes, and they govern histone methylation profiles of a large number of genes that regulate various cellular pathways. So simple thing that is given in your notes, these polycomb bodies, they regulate the gene expression like by through by governing the histone methylation. Well, histone methylation is a process where we can able to regulate the gene expression, right? And that is acting by the polycomb bodies. Polycomb bodies, they act in complex with more proteins, and they regulate the histone methylation of the gene, right, which will ultimately helps in regulation of the gene expression. Okay, so this was about the nucleoplasm. Now I have said about the basic proteins which are found within the nucleoplasm. So generally, broadly, if I say about the nucleoplasm, they are filled with transparent, semi-solid, granular, due to the presence of this type of Kaggle bodies, Gemini bodies, polycomb bodies, granular, and slightly they are acidophilic ground substance of matrix. And generally within this nucleoplasm, it consists of nucleic acids, as we know, and most common nucleic acids are DNA and RNA. Um, and they also contain some proteins like basic proteins. Okay, basic proteins like nucleoprotamines, then nucleohistones may observed. Okay, some non-histone or acidic proteins may also be observed. Some enzymes are also there, which are found within this nucleoplasm. And I think you can able to name that enzymes also, because within the nucleus, what event occur? Replication occur, and for replication, what type of enzymes needed? We know DNA polymerase, helicase, this type of enzymes are there. It also involved in transcription also. So within the your nucleoplasm, it also consists of enzymes like RNA polymerase. Okay, then nucleoside triphosphatase, adenosine diaminase, then pyruvate kinase, then nucleoside phosphorylase, aldolase, enolase. As you have studied little bit in your biochemistry syllabus, different type of enzymes are found within the nucleus, specifically within the nucleoplasm. So I have mentioned some of them. Some cofactors are also there. Some coenzymes are also there, like ATP and acetyl coenzyme A are also found within this nucleoplasm. Lipids, some minerals are also found, but lipids are found in very small quantity, obviously. Okay, so this was about the nucleoplasm. Okay, next we will talk about the chromatin fiber. So now let us discuss about another important component of nucleus, that is your chromatin fiber. Well, chromatin fiber are what? Nucleoplasm. Within the nucleoplasm, it contains many thread-like, coiled, and much elongated structures which actually takes the basic stain if we stain them with laboratory, like the fuchsin stain, FUCHSIN. So it is a basic fuchsin stain. It will take up by these chromatin fibers. They are thread-like, coiled, and much elongated structures. And these are called as chromatin fibers, which are observed during the interphase nucleus. Why? Because when they will involve in cell division, they will be coiled with other proteins. They tightly coiled with the histone and non-histone proteins to form chromosomes. So chromatin fibers are generally observed during your interphase stage of your cell cycle. That is during the G1, G2, and during the G1 phase, S phase, and during the G2 phase is called interphase. But during the cell division stage, that is during mitosis and meiosis, this chromatin fiber become much more thick, ribbon-like structure due to its coiling, due to its condensation and coiling with the other proteins, and form as chromosomes. Generally, chromatin is formed of DNA and proteins. You can see here two types of chromatin here, heterochromatin and euchromatin. Euchromatin is not so much coiled structure of chromatin. It is loosely, you can say a loosely form of chromatin structure, a thread-like structure. And euchromatin structure are mainly observed on those sites where active replication transcription takes place. Because if it is tightly coiled, how will be the enzymes will access to the sites for your replication, for your transcription process. So euchromatin structure is more loosely form of structure, loosely coiled structure, which is observed during the time of replication, which is observed during the time of transcription of certain regions within the chromosomes. The heterochromatin are more coiled form. Heterochromatic regions are more coiled form of structure which are inactive in your replication and transcription process. But when they become more coiled, they become chromosomes. They become thick ribbon-like structures called chromosomes. Generally, these chromatins are made up of DNA and proteins. Small quantity of RNA may also be present, but it accounts for less than 5%. Less than 5% of the proteins are of chromatin are made up of RNA. Okay, some non-histone proteins are also present. Okay, now why we are talking? We have said much more about histone proteins here. We have also said when we are talking about the causal bodies, which is found within the nucleoplasm. Here also we are talking about the histone proteins. Well, histone proteins are the constituents of chromatin that actually helps in coiling of the you can say chromatin fibers to form a thread-like structure like chromosomes. Uh, okay, how they are coiled? If I say in short, well, there are four types of histones which are found. If I talk about histones, it will become a lengthy part. There are four types of histone. Actually, there are five types of histone. Sorry for that. There are five types of histone: H2A, H2B, H3, H3, and H4. and H1. These four types of histone form a nucleosomal complex. Two molecules of H2A, two molecules of H2B, two molecules of H3, and two molecules of H4 together forms a histone octamer. So they together forms a histone octamer, and within this histone octamer, your DNA coiled about 146 base pair of DNA, about 146 base pair of DNA coiled around this histone octamer. So in this way coiling occurs, okay, and it makes more folded structure, okay. Histone H1 associate with this within this histone this empty region, okay, of the DNA. In this way more coiling of the histone occurs, more coiling of this DNA with the histone occurs to form a chromatin like structure, okay. So in this way your histone proteins work. An overview I have said. Generally, two forms of chromatins are observed. One is called heterochromatin and another one is called euchromatin. Heterochromatin, if I say, they are highly condensed region of chromatin, and they are found around the nucleolus. Heterochromatic regions are found around the nucleolus, which I will be going to discuss with you after this. And they are found at the periphery, that means at the end portions of your nucleus. Normally these heterochromatic regions are genetically inactive regions because these regions are not active in your DNA synthesis or RNA synthesis. Whereas the euchromatin regions, as you can see here, right here, they are diffused region of the chromatin, and at this portion your actively synthesis or DNA synthesis, replication and transcription being going observed. Here the histones are not tightly associated with the chromatin protein, but the heterochromatin regions are tightly associated. That's why they are inactive regions. Now let us see about the nucleolus, which is the last part given in your nucleus in your cell biology syllabus. Now, nucleolus is very important because it is the site for the ribosomal RNA production. Inside the nucleolus, as you can see in the figure, RNA molecules are combined with the proteins to form your ribosomes. Nucleolus is involved in rRNA transcription. It is also involved in pre-RNA processing and ribosome subunit assembly. Okay, so nucleus is very important. Why? Because it is a site for the ribosomal RNA production, and within the nucleolus, rRNA molecules are combined with proteins to form ribosomes. Nucleolus is involved in rRNA transcription. It is involved in pre-RNA processing and ribosome subunit assembly. Okay, and nucleolus is not bound by any limiting membrane. Calcium ions are supposed to maintain its intact organization generally, and chemically, nucleolus contains DNA of nucleolar organizer region. Four types of rRNA is also found within this nucleolus. 70 types of ribosomal proteins, then RNA binding proteins like nucleolin. nucleolin. Okay, then all the splicing proteins, RNA splicing proteins, are also observed in this case, where full assembly of the snRNP has been taken place. It also contains phospholipids, orthophosphates, calcium ions. Right, some enzymes are also found within this nucleolus, like your acid phosphatase. It also contains nucleoside phosphorylase, NAD plus synthesizing enzymes for the synthesis of some coenzymes. Okay, then RNA methylase enzyme is also involved here, also being formed here, which also found here, which actually transfer the methyl groups to the nitrogen bases. Okay, of some cells. Within the some cells, we can also find RNA methylase here also, which transfer the methyl group to the nitrogenous bases. These nucleolus are the sites for the biogenesis of the ribosomal proteins, as I have said little bit ago. They are the sites for the biogenesis of the ribosomal subunits, which will form the ribosomes, the 40S and the 60S. Remember, S refers to the Swedberg unit, which actually normally we give this unit according to the sedimentation when it occurs during your density gradient centrifugation process. According to that phenomenon, how they how they settle to the tube after density gradient centrifugation, we give this Swedberg unit term. So the 40S and the 60S ribosomal subunit are the eukaryotic ribosomes, which are normally uh, observed within this nucleolar organizer region, because they are the sites, nucleolus are the sites where your 40S and 60S ribosomal subunit biogenesis takes place. Okay, then it init three types of rRNA. So that within this 40S and 60S ribosomal subunit, it is also ribosomal subunit is made up of rRNA and proteins, like the 18S rRNA, 5.8S rRNA, then 28S rRNA are all transcribed as a parts of a much longer precursor molecule, which undergoes processing here within this nucleolus, right? So many functions are performed by the nucleolus. If I show you the diagram, more about the diagram here, you can able to see. Yes, this is the diagram. Here we can see the three different regions of nucleolus and their involvement in ribosome assembly. If I zoom it out, here you can see. So these are the pre-ribosomal particles which are being assembled. Okay, so this actually takes place within the fibrillar center. The fibrillar center observed within the nucleolus, and it is the innermost region which is found within the nucleolus, the fibrillar center. Well, this fibrillar center that is found within the innermost region of the nucleolus, here transcription of the genes occur. The RNA genes of the nucleolar organizer region of chromosomes are located within this region. So within this fibrillar center, the RNA genes of nucleolar organizer region are of chromosomes are also found. Then there is a dense fibrillar component which is also found within the nucleolus. The dense fibrillar region surrounds the fibrillar center. You can see long strip like uh, lines has been shown. These are the dense fibrillar component. This region, they surrounds the fibrillar center, and RNA synthesis progresses in this region. So in within this dense fibrillar region, your RNA synthesis progresses occur. 70S ribosomal proteins also bind to the transcripts in these regions. Then comes your cortical granular components. Cortical granular components are actually your outermost region of the nucleus, where processing and mature of the pre-ribosomal particles occur. So you can see granular components here. These granular components is the outermost region of the nucleolus, where your processing and maturation of the pre-ribosomal particles occur. So you can see the maturation of the pre-ribosomal particles are occurring. All the other components of the RNA, ribosomal RNA like 5S rRNA, 18S rRNA, then your 28S rRNA are being transcribed here. Okay. So nucleolus, if I say, they are important part. They plays an important role in maintaining the structure. Okay, because they are the sites for ribosomal RNA production. In simple words, and normally these rRNA will be formed, combined to form with proteins to form ribosomes. So within the nucleolus, your rRNA transcription, pre-RNA processing, and ribosomal subunit assembly takes place. Okay. So this was all about the structure and function of the nucleus in detail. I hope you have understood what I have taught to you today. For any problems, you can ask me in the WhatsApp group. Okay, so that's all for today's lecture.